Well, thanks all for inviting me to try to be here. Enjoyed hearing the talks. First, I'd just like to acknowledge my collaborators. Much of the work I'll describe is done with longtime colleagues, Adenosine and Ronald, and also acknowledge postdoctoral research associates Julia, Kumar, Akhtal, and former postdoc Aaron Szymusiak, whose work I'll discuss. So we're back to the big brain, so my lab has long-standing interests in neuronal systems that promote sleep, but in order to understand how close the brain is to sleep, you have to know a little bit about what keeps it awake. And there's good consensus in the field that the generalized electrographic and behavioral arousal of wakefulness emerges from the collective activation of several ascending neuronal systems whose cells of origin are located in the brainstem, hypothalamus, and basal forebrain. So these can include monoaminergic neurons, the locus ceruleus, dorsal raphe, and tuberculous nucleus, and hypothalamus, cholinergic neurons in the brainstem and basal forebrain, and hypocretin neurons, which, in addition to targeting the cortex and the thalamus, also send excitatory inputs to several of the other arousal systems. So this is sort of a modern conceptualization of the reticular activating system, but it's sort of collectively these multiple systems are responsible for the generalized activation of wakefulness. So if you look at the patterns of neuronal discharge among neurons in these different nuclei, as you would expect, since they're related to arousal, they have uniformly have elevated discharge rates or maximum discharge rates during waking, lower discharge rates during REM sleep. Several, like the monoamines and the cretin neurons, are silent in REM sleep, is what the sleep researchers often refer to as the REM-off discharge pattern. Contrast, cholinergic neurons and glutamatergic neurons in the parabrachial nucleus are active during both wakefulness and REM sleep. Of course, the EEG is activated in REM sleep, so presumably they're responsible for maintaining activated EEG during REM sleep in the absence of activity with these other REM-off systems. But the main feature in all these arousal systems is that at the time of the transition from wakefulness to sleep, they all undergo a fairly sudden and dramatic reduction in neuronal activity. In a polyphasic sleeper, like the rat, transitions between waking to sleep occur over a period of seconds. In more consolidated sleepers, like humans, it occurs, if you're lucky, over a period of minutes. So the burden then for a neuronal system that functions to promote sleep has to somehow achieve a coordinated downregulation of these disparate, in terms of location in the brain, arousal systems, and achieve it fairly quickly. So I will try to convince you that one of the ways this is accomplished is by groups of sleep regulatory GABAergic neurons that are located in the preoperative hypothalamus. So just as arousal promoting neurons can be identified by their elevated discharge during aroused states, sleep promoting neurons are candidates for sleep promoting sleep regulatory neurons to be identified by their activation during sleep compared to waking. And so we initially studied this in cats and rats using extracellular unit recordings with the chronic microwires and understanding naturally sleep-waking rats. And this is an example of what we would call a sleep-active imputed sleep regulatory neuron, which shows elevated discharge during periods of waking and reduces discharge during periods of sleep. One of the places we most commonly encounter these types of neurons, shown here in red, is in the preoptic region of the hypothalamus, where we would find sort of a dorsal distribution also with the anterior commissure, and a more ventral distribution lateral to the optic chiasm. When you sort of look at the discharge of these sleep-active neurons, see they have low discharge during waking, elevated discharge during both non-REM and REM sleep. It's the exact reciprocal state-dependent discharge pattern that you see in the REM-off neurons typified by presumed histaminergic neurons in the tuberculous nucleus, and presumed hypocretin neurons in the lateral hypothalamus. The other way you can identify the these sleep regulatory neurons is by looking at sleep-related CFOS expression. This was initially done by Cliff and Ronald. They showed that they found a ventral cluster of neurons that express CFOS during sleep, but not during waking, located in the ventrolateral preoperative area here. These three grids are typical counting grids that we use to quantify state-dependent FOS expression in this area of the brain. We identified a second area corresponding to that dorsal cluster of sleep active neurons we defined physiologically, located in what roughly corresponds to the median preoptic nucleus, which is 3D and NPO. These are the sort of standard counting boxes that we use to quantify FOS expressing cells in the rostral versus the caudal components of the NPO. We were able to show that the sleep-related FOS in both the NPO and VLPO is co-localized with markers of GABAergic neurons, and that's shown over here. We're in a fairly large group of spontaneously sleeping rats. Some are poor sleepers, they have low sleep efficiency. Some are good sleepers, they have high sleep efficiency. The time spent asleep in the 90 minutes prior to being killed is correlated with the number of GABA neuroreactive neurons expressing FOS. The safer group has shown that in the VLPO, the sleep active cell groups, the normal group co-localizes both GABA and neuropeptide alanine. That's not the case for the neurons in the NMPA. They don't seem to express alanine. Originally, the work from John Sharon in Cliff Safer's lab identified the sleep active neurons of ventrolateral preoptic area as being source of descending projections. Specifically, sending a very dense projection to the histaminergic region in the tuberomammillary nucleus. That's really how that nucleus first caught their eye, was from that anatomy. It also projects to the other monoaminergic subgroups as well as cholinergic neuronal groups in the brainstem. We, postdoc in my lab, Aaron Szymusiak, a few years back, looked at the projections from the median preoptic nucleus, that dorsal cluster of sleep active neurons. Here using biostimulated dextran and anterograde tracer, demonstrating positive axons in the hypocretin neuroreactive neuronal field. Here's an instance where the axons are making sort of close positions to hypocretin neurons. Here in dark field, demonstrating projections to the dorsal raphe nucleus and the locus ceruleus. So the median preoptic nucleus projects to some of the same places, into potentially some of the same arousal systems as the VLPO. Aaron was able to show that a component of these descending projections exhibited sleep-related CFOS activity. So here, for instance, he performed injections of the retrograde tracer fluorospheres into the paracortical area in the region where hypocretin neurons are located. It's retrograde labeling throughout the preoptic area, including the VLPO and NMPO. And he did this in a series of animals, some of which he would kill after a period of wakefulness, some of which he would kill after a period of consolidated sleep. And what you can see is that here in the awake animal, there's retrograde labeling, the VLPO, but very little FOS. Here in the sleeping animal, you can see co-localization of FOS. And red fluorescence, same thing in NMPO. So there's a series of studies like this performed in my laboratory and in other laboratories. The idea has come about that a mechanism is responsible for the transitions from waking to sleep, or as Cliff Safer likes to call this, the sleep state switch between waking and sleep, arises due to the activation of these GABAergic, in the case of the VLPO neurons, GABAergic gamma-energic neurons during transitions to sleep and during sleep, causing GABA-mediated inhibition of several arousal systems. So waking to a relatively compact group of sleep active neurons can influence the activity strongly and quickly in several different key arousal systems. So while certain aspects of sleep regulation, like the transition from waking to sleep, are sort of switch-like, there are also longer time courses process S, accumulates as a consequence of prior time awake, and that when the circadian gate opens, permits sleep, and process S is high, then you have a short latency expression, consolidated period of sleep, during which time process S deteriorates or internalized, or you don't want to think about it, and the cycle starts all over again. So this is a human who sleeps during the night. This is drawn here. Again, we're talking about rats. Remember that they're nocturnally active and they sleep during the light phase. So what's the biological basis of process S? Well, I'm going to be there that the answer cannot be given definitively. It's probably a complex answer, but certainly people speculate that there may be some kind of neurochemical, metabolic process regulating this. Because we know a lot about process C. That resides in the clock in the brain, the suprachiasmatic nucleus. We're less certain about process S, but certainly some kind of endogenous sleep factor, neurotransmitter, neuropeptide, neuromodulator that accumulates during waking and declines during sleep is an attractive candidate. And so there are probably 4 or 5 sleep factors, including some cytokines, which there's good evidence that they play a role in sleep homeostasis. But by far the most convincing evidence for an involvement is adenosine. So adenosine, of course, is a metabolic byproduct of ATP use and degradation. So the relative ratio of ATP to adenosine is a good indicator of brain energy charge. There are enzymes that convert ATP to adenosine located intracellularly and extracellularly, and there are transporters that can move adenosine in and out of cells. Importantly, glial cell, our astrocytes in particular, are an important source of extracellular ATP, probably the major source of extracellular ATP, and that can be converted extracellularly to adenosine. And there are studies I'll summarize briefly that indicate the adenosine source from astrocytes is important in homeostatic sleep regulation. So this is just a brief, and by no means complete summary of some of the types of evidence implicating adenosine sleep homeostasis. Adenosine agonists increase sleep, and they increase EEG slow wave activity during sleep. So elevated slow wave activity during sleep is a reliable biomarker of a homeostatic sleep drive and sleep debt. More about that. Adenosine antagonists, caffeine, promote arousal. In certain brain regions, extracellular levels of adenosine increase during sustained waking and decrease during subsequent sleep. Conditional knockout or knockdown of A1 adenosine receptors in the cortex can blunt the EEG slow wave response during recovery sleep following sleep deprivation. I mentioned before, we block ATP release from astrocytes and disrupt homeostatic responses to sleep deprivation. It seems that both inhibitory A1 adenosine receptors and excitatory A2A adenosine receptors are involved in sleep regulation. So A1 receptors are located throughout the brain. They have inhibitory effects on a variety of cell types, including several that are implicated in arousal and control of EEG. The A2A receptor also appears to play a role. The alerting effects of caffeine are dependent upon intact functioning A2A receptors. And there's some evidence that A2A agonists promote sleep loss expression in VLP neurons. So one factor that might link the sleep active neurons in the preoptic area to sleep homeostasis is this responsiveness to adenosine via the A2A receptor. So we conducted a detailed examination of how A2A receptor agonist affects FOS expression in GABAergic neurons and NMDO and VLPO, and that had never specifically been looked at. We looked at we delivered the A2A agonist by 2 routes of administration. One into the ICV, into the lateral ventricle, and the second is in this, actually delivering into the subarachnoid space just rostral to the hypothalamus or basal forebrain. That's SSRBF, subarachnoid space rostral to the basal forebrain. So why deliver it there? In part because other groups, Hayashi's group, has shown that other sleep-active substances, when delivered into this region of the subarachnoid space, in particular prostaglandin B2, are most effective in promoting sleep. And if you look at the configuration of the location of the median preoptic nucleus here, and then the ventrolateral preoptic nucleus here, just lateral to the occipying at the base of the brain, they're both located very close to this region of the subarachnoid space. So this is potentially a very site that could very efficiently deliver drug to target these neurons. So this is the sleep results. So this is the CGS26180 is a selective A2A receptor antagonist. And here in the ICD group, we gave 2 doses, 8 nanomoles, 24 nanomoles in the vehicle. In the subarachnoid space, we just gave the vehicle an 8 nanomole dose, and as we predicted, you see a significant decrease in waking and an increase in sleep in response to the ICD administration of a higher dose, whereas the 8 nanomole dose, which is not effective when delivered ICD, produces significant decrease in waking, increase in non-REM sleep. So we're going to look at FOS expression and our nuclei of interest. Here we're looking at the percent of GABAergic neurons that are double labeled for CFOS, and that increases in a dose-dependent manner in the for ICD group in both components of the median preoptic nucleus and the so-called core and extended ventrolateral preoptic area. And importantly, we did a third experiment where we gave the 8 nanomole, here's the 8 nanomole dose in an undisturbed animal as the animal is allowed to sleep. You see increases in FOS expression. And here, we kept the animals awake. So we didn't allow them to sleep following administration of the drug. So even though there's no sleep being expressed, we still see the increase in FOS expression, which suggests that, which indicates that the A2A activation can excite these cells in a state-independent manner. So the CFOS, in terms of studying these sleep-active cells, the CFOS has certain advantages, principally because it gives you kind of a functional and anatomical perspective, which is nice, and it also allows you potentially to identify the transmitter phenotype of the cell, the cells of interest. But it's terrible for looking at dynamic responses, either to changes within states or to changing, rapidly changing things like homeostatic sleep pressure. So we really wanted to look at this at a single neuron level to determine, as we would predict from the model, that the neuron that is responsible for regulating homeostatic sleep drive should show dynamic changes in neuronal discharge during accumulation of homeostatic sleep pressure, during wakefulness, during the deprivation period, and during and decline in discharge rate across recovery sleep. Now, practically speaking, it's not possible to do this for a 24-hour period because it's really difficult to maintain the recording of a single cell for that period of time. So what we did was to study. We're now looking at rats. We're studying rats at this point in their curve, where they've had a high degree of sleep pressure because they've been awake during the evening, and the circadian gate is open to permit sleep. So, for instance, if you are a habitual sleeper and your bedtime is delayed, you don't have to have a long period of sleep deprivation to become very sleepy. So I'm a habitual early riser, and if I get pushed much beyond 9:30 or 10 at night, I don't do very well. It's evidenced by the fact that I really wanted to watch the Dodgers clinch the NL West against the Giants last night, and I was very motivated to do that, but I fell asleep and had to check off. So essentially, we're doing that to the body, and I'll show you what I mean by that. So this is the experimental paradigm. So So we began recording, searching for neurons and recording right after lights on. So that is the sort of peak time of endogenous sleep pressure in the rat. It's been up all night, or much of the night. It's exactly phased. And now the circadian gate is open to allow sleep. It has a high level of spontaneous sleep drive. So we identified the neuron during a baseline recording period as being sleep active. So here's a period waking. Here's discharge during sleep. Here is when sleep deprivation begins. And so you can see that the cell is more active during sleep than during waking. So the sleep deprivation starts here, continues for 2 hours. Each one of these little tick marks here indicates when the experimenter, who is watching the rat, notices that it's attempting to fall asleep, either behaviorally or through some changes in the EEG. And it has to then, the experimenter then has to intervene with what sleep researchers euphemistically call gentle handling to keep the animal awake. And this involves either tapping the cage, touching, stroking the fur of the rat with a cotton swab or something, just to cause a brief arousal to keep it awake. And so what you can see is that when you first start to sleep deprive the animal, the experiment's not work that hard to keep it awake. It only has to intervene occasionally. But as the sleep deprivation continues, you see this progressive increase in the number of sleep attempts, and by the end of 2 hours, the experimenter is having to intervene almost continuously. That's the point at which the gentle handling might get not so gentle. But this gives you a nice measure, simple measure or biomarker of increasing sleep pressure during the sleep deprivation period. All right? And then at this point, the animal is released into the recovery state. So what you can see in terms of the discharge of the neuron is that during this period where the behavior indicates that homeostatic sleep pressure is relatively low, the cell fires at relatively low discharge rates, comparable as it does to baseline waking. But then, as the deprivation period continues, discharge rate increases such as that by the end of the deprivation period, the cell is firing faster than it did during baseline sleep. When you release the animal to recovery sleep, you initially see that those high discharge rates are maintained, but then they decline back towards baseline levels as the recovery sleep period continues. And this just shows a higher expansion plot here for B, early in the deprivation, here for C, late in the deprivation, change in discharge rate. And note, this increased activity is not the result of the rat having little micro sleeps. They do have little micro sleeps, but we can count the discharge rate during periods where they're fully awake by EEG and behavioral criteria. These are superimposed action potentials recorded from the different sections of showing to convince you that this is a stable unit recording, and these changes aren't due to changes in signal. So this is mean data for 11 sleep-active neurons in the median group that localized in the median preoptic nucleus during this paradigm. And what you can see is that when you look during the sleep deprivation period, there are significant increases in the mean discharge rate of these neurons late in the deprivation period compared to early in the deprivation period, and they perfectly correspond with this increase, the significant increases in our behavioral metric of sleepiness, attempts to fall asleep. During recovery sleep, you can see that both discharge rate and EEG slow wave activity, our biomarker of homeostatic sleep pressure, is higher than baseline early in the recovery period, but then declines as recovery sleep continues. So the cell then looks very much like it is responding in a very dynamic way to minute changes in homeostatic sleep pressure. So putting these findings together with what we found with our drug infusion studies of A2A adenosine receptor agonists, you might predict that what is responsible for these increases during sleep deprivation here is elevated levels of adenosine acting on A2A receptors and activating cells. So to try to address that question, we used a technique that was developed perfected by Ronald in our group, which is to combine drug delivery via reverse microdialysis with single unit recording. And so it involves essentially planting 2 guide cannula, 1 fixed, to guide a microdialysis probe, 1 on a movable microdrive holding a bundle of microwires. So when we're ready to do the experiments, we insert the dialysis probe in place, and then you have like 3 or so days to do your neuronal recording while the dialysis membrane is still functional and is not corrupted by gliosis or other factors, and then you just make a microwire pass along the length of the dialysis membrane, so you're recording within the field of drug delivery by the dialysis probe. So we did this in the ventrolateral preoptic area. Practically speaking, this method does not work for the medium preoptic nucleus because of its midline location and its size. So these are just a little more accessible effectively. So this then shows here in the baseline period, just like the experiment I showed you before, in the baseline period, we identified the cell as being sleep-related. Here we start sleep deprivation. Initially, the discharge rate is low. Late in the sleep deprivation period, discharge rate increases, as reflected by increasing homeostatic sleep drive. And then we give a 20-minute infusion of this A2A adenosine receptor antagonist here, which results in a fairly rapid decrease in discharge rate that is actually sustained for about 80 or so minutes throughout the recovery sleep period until eventually you see recovery of discharge. And this just shows here in B, in the baseline period, the difference in discharge between waking and non-REM sleep. Here, early in the recovery period, discharge during waking and non-REM sleep. Here's the demonstrating stability in unit across the paradigm. So we did this. These are very difficult experiments. A lot of these have to go right, so the ends are small, but the data are convincing that we had a group of 6 sleep apnea neurons that we studied this way, and comparing vehicle to A2A adenosine antagonist diffusion, there's significant decreases in both states in response to the drug. We looked at a small population of wake-active neurons, and they showed no effect, or no response. So this suggests then that sort of part of the normal homeostatic response to sleep loss involves an activation of these sleep regulatory neurons in the preoptic area, mediated in part by elevated adenosine acting via A2A receptors. So the last thing I want to talk about is just some recent work we're doing looking at corticotropin releasing factor in the context of sleep regulation and sleep homeostasis. So it's well known that CRF promotes arousal and suppresses sleep as part of the stress response. That's been known for some time and quite well characterized. In fact, it's known that CRF-induced arousals are mediated in large part by excitatory effects on noradrenergic neurons, locus coeruleus, and hypocretin neurons in the lateral hypothalamus. So I'm going to show you some sort of work in progress demonstrating that CRF appears to be, in addition to causing sort of straightforward arousal effects, appears to be a negative modulator of homeostatic sleep regulation occurring in response to acute and chronic sleep loss. And in addition to effects on arousal systems, CRF may also interfere, particularly under chronic conditions, with sleep regulatory functions of these DLQ and NPO neurons. So initially we did an experiment looking at just the basic effects of ICV infusion of CRF here in a group of rats, including CRF and 6 here in the group, just in their home cage. And this is percent time awake, non-REM, and REM sleep. We did a dose response curve, which I'm not showing you, but I'm showing you this 0.1 nanomole dose, which gave us sort of the threshold dose in our hands. That is, we got suppression, an increase in waking, suppression of non-REM sleep for the first 2 hours after the ICV injection. There's a trend sort of decrease in REM sleep time, but it wasn't significant. Again, higher doses would give more severe sleep disrupting effects and also affect REM, but we were interested in comparing the effects of this sort of intermediate dose, if you would, on the sleep response occurring when animals are exposed to a mild stressor. So what it shows is this sort of a mild social stressor, which involves involves exposing a male rat to the dirty cage of another male rat. So it's a sort of a common type of stressor that people will use, for relatively mild, but it's clearly something that the rats don't like. And you can see here in the vehicle-treated rats, they initially respond with actually nearly complete absence of sleep, and compared to home cage controls, they have some decrease in sleep time that persists about the first 3 hours of exposure. Whereas in response to the CRF, the one animal dose, same as here, there's a tremendous potentiation of the sleep disruptive effect of this mild stressor in the presence of elevated CRF signaling. So you see this increase in waking, decrease in non-REM sleep that lasts for 4 hours, as compared to 2 in the home case. Really a dramatic change in the effects on REM sleep, where you have the suppression of REM sleep lasting out to 7 hours. So this clearly has the CRF then potentiates the sleep suppressing effects of this stressor. And the second group of 6 rats exposed to this paradigm, vehicle and control in the cage exchange paradigm, we killed them at hour 5, post CRF injection and cage exposure, and looked at FOS expression. And what we found were large decreases. So even though at 5 hours, while the REM sleep deficit persists, much of the non-REM sleep deficit has recovered, but we still see large decreases in the number of GAD neurons expressing FOS in both the NNPO and the VLPO. We did another sort of straightforward probe for how CRF impacts homeostatic sleep regulation, where we deprived animals, rats, for 3 hours early in the life phase, gave them ICV injection of CRF during that sleep deprivation period, and then looked at recovery sleep. Here, 90 minutes of recovery sleep after which we killed the animals to look at FOS. And what you can see is that the CRF infusion causes suppression of all the components of the homeostatic response. There's an increase in time spent awake in CRF rats. There's a decrease in the time spent in non-REM and REM sleep. There's an increased latency of the first non-REM sleep episode. The mean duration of non-REM sleep episodes is decreased latency of the first REM episode. REM bout duration is decreased, and there's a decrease in EEG slow wave activity during non-REM recovery sleep. And this then, looking at the FOS, is associated with decreased FOS expression in GAD neurons in both components of the both sleep regulatory nuclei and the preoptic area. So again, this is sort of evidence that CRF has sort of negative modulatory effects on sleep homeostasis. So now, giving the drug ICV, you're delivering it everywhere. You have no idea how direct the effects might be on the preoptic sleep regulatory neurons. Part of this really all could be secondary to drug-induced state changes rather than direct effects. So we attempted to try to get a handle on that by doing infusions of CRF receptor agonists, CRF receptor 1 agonists, stressin, into the subarachnoid space, adjacent to the sleep regulatory nuclei. And so we did 2 doses, 0.3 and 1 micrograms, delivered during a 2-hour infusion period. So this is the total amount delivered during a 2-hour infusion. And so this is the sleep amounts. This is wake, non-REM, and REM. So there's a trend for dose response for both awake and non-REM sleep, but the significant difference is only at the high dose. There were no significant changes in REM sleep. So then we looked at CFOS expression in the 3 conditions, vehicle to 2 doses of drug, and VLPO. We compared it to FOS expression in hypocretin neurons in the paraformical area, decided by CRF. And what you can see is that the high dose, indeed, while the sleep is decreased, FOS expression is decreased in NMP and BLPL and increased in the hypocretin neurons. But at the low dose, there's a significant reduction of FOS expression in medium preoperative nucleus neurons, even though there's really only modest changes in sleep, and there's no effect on the hypocretin group. So this is sort of evidence that suggests perhaps you can get relatively direct effects of CRF neurons on the sleep active neuronal component without having big changes in behavioral state or global activation of arousal systems. So the last point I'd like to make, or thing I'd like to discuss, relates to effects of chronic sleep restriction on sleep homeostasis. So up to this point, we're looking at relatively short-term periods of sleep deprivation, and on our hands, 2 to 3 hours delivered during the times of high intrinsic sleep intensity, so you get a strong homeostatic response. An decrease in sleep latency, an increase in sleep amount, increase in sleep consolidation, increase in EEG slow wave activity during sleep. Now, when you do a more chronic partial sleep deprivation or sleep restrictions study, and several labs have done this, this was first done in Fred Turk's lab, what you see is that initially, so the original paradigm was to restrict sleep to a single 6-hour period during the day and deprive the animal for 18 hours. So when you do that, the first day of that paradigm, when the animal has the opportunity to sleep, it shows a strong homeostatic response. It shows increase in sleep amount, EEG slow activity, sleep consolidation. But if you continue that for days, eventually the homeostatic response disappears. You don't see changes in sleep. You no longer see the increase in EEG slow activity when the animal has an opportunity to sleep. So this has been viewed as kind of a sort of allostatic adaptation to continued partial sleep loss, which changes the way the system tries to deal with the stress of sleep loss. And there's no question that decreases in sleep are an appropriate response to certain kinds of stressors. But chronic stress can lead to impaired sleep, and that seems to be the case in rodent models that the chronic repeated partial sleep loss causes this weakening of homeostatic response. So we wanted to look at this in the context of, well, is there a role for CRF signaling, changes in CRF signaling in these changes in homeostasis with chronic sleep restriction? So we, rather than use the 6 hours of sleep, 18 hours of forced wakefulness paradigm, we adopted a paradigm that was originally published by Kathy December a few years ago, where it's a 3-in-1 paradigm, where you have periods of 3 hours of sleep deprivation followed by 1 hour of sleep opportunity, and you have that throughout the 24-hour day, so you still have 6 hours of sleep opportunity. It's just divided up into separate terms, separated by these periods of deprivation. In some ways, it seems to be a more viable strategy for looking at a polyphasic sleeper like a rat, who doesn't have consolidated sleep. So forcing to sleep in one consolidated period may not be quite so appropriate. And just the way sleep. So for this experiment, the animal lives on a treadmill. He's got a cage. Before the cage is a treadmill. He's got a water bottle. He's got everything he needs. It's just that during these 3 hours of sleep deprivation, every 12 seconds, the treadmill belt turns on for 3 seconds. So the animal has to step. So he can't. They get almost virtually no REM sleep, and they get very little non-REM sleep. But non-REM sleep they get is very fragmented. And we've used this paradigm before to produce total sleep deprivation. But here we're using it to produce chronic sleep restriction. And so what we did in this experiment was on day 4, when the allostatic response is apparent, that is, the animals are no longer showing a homeostatic response during the sleep opportunity, we gave, during the 3 hours of deprivation, we gave infusions of CRF1 receptor antagonist antialarmin, and it's 10 micrograms was the total amount delivered over the 3 hours. We did that for this deprivation period that ends at ZT5. So ZT is Zeitgeber time, ZT0 is the time of lights on. I'm going to do it for the second one that ends at Zeitgeber time, or begins at ZT9, I think it is. All right, and then we've looked at the recovery sleep measures here and compared to the other days, including a day 5 that we did with the drug. So here's what we found. In terms of EEG slow wave activity, expressed as percent baseline, this is EEG slow wave activity in non-REM sleep, our biomarker of homeostatic sleep drive. Here's baseline. On the first day of chronic sleep restriction, there is an increase in EEG slow wave activity during non-REM sleep that declines on day 2 and is back towards baseline by day 3. On day 4, when we do the infusion of CRF receptor antagonist that restores the EEG slow wave response during sleep, it does it for that sleep opportunity ZT5 and ZT9. Here is the non-REM sleep alterations. So I'm showing you baseline, CSR1, CSR3. I'm not showing you day 2. And here's day 4. So you can see that there's a big increase in valve durations on the first day. After the first day of sleep restriction, it is back to baseline by the 3rd day, and the infusion then of CRF1 receptor antagonist restores that aspect of the homeostatic response. So this suggests that part of this suppression of the homeostatic response is in response to CRF, increased CRF signaling, due presumably to the chronic mild stress of this sleep restriction paradigm. So just to briefly summarize then, I've convinced you that activation of VLP-IMP GABAergic neurons is an important critical step in orchestrating orderly transitions from wakefulness to sleep and also for maintaining normal sleep. That the normal homeostatic response to sleep loss depends, at least in part, on adenosine-mediated activation of GABAergic sleep regulatory neurons in the preoptic area. That CRF appears to be, in addition to, in acute situations causing, hyperarousal, which is an appropriate component of stress response, also seems to be a negative regulator of sleep homeostasis, particularly in chronic sleep restriction situations, and that some of the adverse effects of stress on sleep regulation may be due to CRF-mediated disruption of the sleep regulatory neurons and circuits in the preoptic area. So I have some time for questions.
Questions? I apologize if I missed it earlier in your talk. What happens if you lesion these areas?
So ventrolateral preoptic area lesions cause fairly dramatic, persistent deficits in sleep loss, and median preoptic nucleus also has been shown to produce more modest decreases in sleep. The problem with the median preoptic nucleus is that it's involved in several important things, including body fluid homeostasis, thermoregulation. So if you make a large lesion there, you get a very unhealthy animal. It's difficult to interpret the sleep effect. But sort of smaller lesions lead to some modest reduction in sleep. Yes?
You had a slide where you had an adenosine antagonist that you had used, or was it agonist?
Well, we did the first study with the dialysis. It was an antagonist, so that was in the context of sleep. The increase in neuronal discharge in response to sleep deprivation was diminished by local infusion of A2A antagonists.
So the animal still went to recover sleep?
Yes, because it's kind of a delicate dance when you do these reverse dialysis drug infusions. So if you do it unilaterally and you modulate the dose and you do a relatively brief infusion, you don't get a global state change. But you can with, obviously dose and duration of infusion. If you do it bilaterally, you can get behavioral changes. So it's a nice thing, though. It allows you to dissociate the global state from the discharge of the neuron, hopefully by somehow altering the level of adenosine signaling in that relatively restricted area where you're recording. Yes.
Thank you for the talk. I have a question about the CRF story. So, seems like the CRF was quite acute effect, except for the REM sleep. Do you think there's like the HPA axis co-activation? So the CRF is only the starter, and like other stress hormone like glucose.
Oh yeah, no, absolutely. Giving the exogenous CRF is going to have an impact on HPA axis activity. And so actually, one of the things that we're doing now in this chronic sleep paradigm is looking at ACH and court peripherally during the days where we do and don't infuse CRF, because part of this might be mediated, correlated with changes in HPA. It's definitely an issue that needs to be addressed.
Thank you very much, Ron. I want to thank all of you guys for sticking out the long afternoon. I also would like to thank a couple of people, Mark and Sarah, and Stacy and Art, for helping put the symposium together, putting together the flyer. If you see them out there, please congratulate them on a nice job. And tell Josh that I didn't thank him for his eye candy, which was the little bar at the top of the poster. So thank you very much. And as Mark said, let's have a symposium.